Hi guys, how are you guys doing? Hope you guys all are doing really great. Today I'm gonna make a video of the physiology of second year, and the topic that today we're gonna be doing is the special senses, and the topic of it is the optics of vision, the eye. Optics is always that field that is related to eye. So in this chapter, what we're going to study is all about the eye, the parts of the eye, everything. All right. Right now, our first topic is the optics of the eye and the eye as a camera. Now, you see this figure, right? This is very important to understand. For example, you have an object placed here. When that object will spring, or when this eye will see that object, what it will do is that it is has to reach here in the retina. So to make the object reach the retina, it has to pass through these different layers and then finally reach the fovea of the retina where the image is gonna be formed. Now, there are four different layers from where the object has to pass to reach the fovea of the this retina. First is gonna be that the object, it is gonna give its rays. That rays is first gonna pass through the air that is present, this one, and the anterior surface of the cornea. So it is gonna pass between these two. Then it is going to pass between this posterior surface of the cornea, and in here you have a aqueous humor present. So it is then going to interface between the posterior surface of the cornea and the aqueous humor. After passing through the aqueous humor, it is going to enter into this lens. It is going to interfere between this posterior surface of your aqueous humor and the anterior surface of the lens. Then last, it is going to interface between the posterior surface of the lens and the vitreous humor. And then finally, it is going to reach the fovea of the retina and where the image is going to be formed. Now, its normal value of diopters is 20 diopters. That is the normal range of the diopters of the image that is formed here at the fovea. Now, how an image is formed in the retina is basically upside down, which is explained here. A person is like this. He is playing a cricket ball, right? The image, we see a person, that image comes into our eyes, same passes through the air, then the aqueous humor, then the posterior surface of the cornea and the aqueous humor, then the posterior surface of the aqueous humor and the anterior surface of the lens, then from the posterior surface of the lens to the vitreous humor, finally reaching the fovea. Now, once it reaches the fovea of the retina, this picture is going to be upside down, it is going to be inverted. Means, if for example I'm seeing this pencil, now this pencil when reaches my retina of the eye, it is going to be like upside down, like this. Why it is so? Because our brain is trained to pick up the signals of the images upside down. So once the brain reaches this upside down image, he will be able to detect and make the motor senses go to the eye, and the person will be able to see normally. But if a person is not able to see upside down image, the brain will not be able to detect the object, and as a result, the person will suffer from different diseases related to the eye. Okay? Alright, now our next topic is the accommodation. Right? What is an accommodation? It is basically in this process that, wait, let focus. Yeah, here. Accommodation is basically that we just said that the normal amount of the diopters in an eye is your 20 diopters. Now, and the eye lens is basically a convex. Now, if instead of 20 diopters, its value increases more than 20, let's say up to 34, then we will say that due to this increased value, the lens has been accommodated by your ciliary muscles. And this process of increase in the value of the refractive index and diopters from 20 to 34 is called as the accommodation of the lens. Now, you see in this picture, you have this lens, and just attached to the lens, you can see these are your suspensory ligaments present right next to the lens. Now, what is the function of these suspensory ligaments? That these suspensory ligaments, once attached to this lens, will either cause the lens to move in your medial direction, in the lateral direction, in the superior direction, and in the inferior direction. So So whenever, for example, I am looking at this object, right? For example, this pencil is my object, and I want to look at it in the frontal direction. So obviously my superior muscles will be contracted. The suspensory ligaments will contract and move the lens in the upward direction, and we will see the object here. Now, if the image is downward, now my lens has to see downward. So obviously these suspensory ligaments will make my lens move in the downward direction, and I will see the object. If this object is placed in the lateral direction, then this lens, for example, this mobile camera is my lens right now, it will move in the lateral direction by the contraction of your suspensory ligament, and we will see the object. If this object is present in the medial direction, then the suspensory ligaments will contract and cause the lens to move in the medial direction, and as a result, we will finally see your image. That is called norm. Now, when we can see here, you see this is the lens. Now, instead of these suspensory ligaments, the lens, they are also surrounded by another layer of your smooth muscle. That second layer of muscles is called as the ciliary muscle. Now, what does the ciliary muscles do? They are of two types. Number one is the meridional fibers and the circular fibers. These together will contract with the suspensory ligaments. So, for example, let me show it. If this is my ciliary muscles, right? This, for example, is my suspensory ligament, and this is my lens. It's right now here. Now, when this will contract, obviously, this is going to move inward, and my convex lens will become deeper and deeper, and finally, it become a deep convex lens. So, this is known as accommodation, and this is used only when we want to focus very, very close objects, right? Then accommodation is basically done. Next comes the nerve innervation. Nerve innervation of this combination is done by the parasympathetic nerve. The parasympathetic nerve fibers of the oculomotor nerve, which is your cranial nerve number 3, will get stimulated, which will be stimulated when we see an object. It will take the impulses from the eye through this hole forming at the fovea of the retina. Parasympathetic nerves will be stimulated. They will take the impulses from the retina all the way by passing through your optic chiasma to your optic tract. From there, it enters the brainstem all the way up by means of the optic tract to reach your occipital lobe for the visual cortex, and then the image will be formed, and the brain will become aware, and we will see the image. Right? That was all for your accommodation process. Okay? Now we have a disease pathology that is called as your what's that word called? It's presbyopia. Presbyopia. Now, what is a presbyopia? It is that we have seen here that basically you have the lenses, and you had these suspensory ligaments, and you have followed by these your ciliary muscles. Now, these ciliary muscles must have some nutrition. The muscles must be provided with some proteins. Now, if these muscles are not provided with proteins, what will happen? Obviously, the muscles will become weak. When muscles become weak, they will not be able to perform their function. Now, if ciliary muscles do not get energy, what will happen? They will collapse. They will become weak. And once become weak, they will not make the focus clearly. They will not cause the convex to become more sharp. The person will then fail to see an object near to him or far to him. And the person will suffer from a pathology that is called as the presbyopia. All right? Okay. Then we have certain diseases that are here that is related to our errors of refraction. In errors of refraction, we have different diseases. Let's start first with what is the normal, and then we will go into the different diseases. The normal, you can see here is, let me focus it. It's called as your emmetropia, which is the normal vision. Right here, they have shown to you the different valves. First, we're going to see the emmetropia, which is this figure. What happens here is that the ciliary muscles, they are going to be continuously relaxed. It is going to be the suspensory ligaments and the lens which will function. Here what happens is that once we see an object, like for example, I see this object very close to me, right? This is present here. Now this will then pass through the air, through the aqueous humor. From there it will pass through the lens, through the vitreous humor, finally reaching the fovea. Once it reaches the fovea, it is gonna come enter into the lens. On entering the lens, it will converge, and then it will pass into the vitreous humor, and then these all lenses will finally meet at a single point at the fovea of the retina, and then image will be formed. This is in case of normal person, and this is called as your emmetropia. Now, if you have a pathology of the eye, what will happen? Two types: hyperopia and myopia. What happens in hyperopia? Hyperopia is a pathology in which your ciliary muscles are relaxed, but the eyeball, your lens, it will become too small, and the lens system will become weak. Now, once the lens system is weak, what will happen? The convergence, the divergence of the parallel rays from the object will not function normally, and they will function at a later stage. So if they had to converge here, they will not converge here, they will converge here and form an image placed ahead of the fovea of the retina, and the image will be formed ahead of the retina, and the image seen by the person will be blurred. And this pathology is called as hyperopia. Now, once the lens is too big, you can see this circle is way too big than this circle that is present in the hyperopia. Then in what this happens is that your lens system works too strongly and your eyeball is too big, and the images of objects raised, they will converge at a point before that of the fovea of the retina, and here also the image form will be blurred and unclear. Now, whenever there is a pathology, there is always a treatment. Now, what is the treatment of the hyperopia and what is the treatment of myopia is shown here in this figure. When a patient has a myopia, means the eyeball is too big, your lens system is too strong, image is formed before that of the retina, we have to make it form at the retina. Then for that we will use a concave lens. This concave lens will be present. This will cause the rays coming from the object to diverge, and they will diverge and they will normally pass through the lens, and they will diverge and they will meet with one another at a point at the fovea of the retina. Now, when a person has a deficient disease or a pathology of hyperopia, means that the image is formed beyond that of the retina, so we want to make it form at the retina. In that case, we will use a convex lens. In using a convex lens, the light rays will pass through it, they will converge, and they will meet with one another at a point at the fovea of the retina and not beyond that of the retina. So this was how you treat patients with myopia and how you treat a patient with hyperopia. Now we have a disease that is called as your astigmatism. Now in astigmatism, you have these different points A, B, C, D. And all of these, they are responsible to pick up your light rays coming from an object. If the image object rays picked up by the object from point A and C is this way, and for B and D is at right angle, like they're perpendiculars with one another. But if the light rays coming from A and C and B and D, they do not focus at the fovea of the retina, they focus at different point. Like if A and C is focusing at here and B and D is focusing here, obviously the light rays, they will not converge at one another, they will not form a single image at the fovea of the retina. Image will be will not be formed, and you will be seeing a blurry image. And this type of disease is called as the astigmatism. Right? And how you treat astigmatism? And astigmatism is treated by putting in cylindrical. When you will put cylindrical lenses like these cylinders, which I think they've shown to you in the next page. Yeah, like here. These cylinders have been produced. So when you will place these cylinders one by one, and you will continue to put these cylinders into the lens system of the eye, so until that image is formed at the fovea of the retina. Once it has been put, then you will not longer put any more cylinders in. Okay, and the other method is by a formula, which is not for your levels. That is for little like who wants to do masters in optics or everything. That is for them. Okay. Then we have a thing called as the cataracts. What is a cataract? The cataract is basically that area. We said that this one